Good afternoon. I'm Narjes. I'm a medical doctor and also PhD student of High Field Research Group at Department of Radiology of UMC Utrecht, the Netherlands. And I'm going to talk about the metabolic imaging of neuro tumors, the MIN trial. As you've heard on the previous presentations, glioblastoma is the primary malignant brain cancer in adults. And as it's the most aggressive brain cancer, it has a poor prognosis with a median survival of only 12 to 18 months. And metabolic reprogramming is the central characteristic of cancer.
That's scary.
And conventional MRI actually gives valuable structural and anatomical information about the lesions and is used to monitor patients before, during, and after treatment, but the information is not at biological level. So nowadays, actually, invasive tumor biopsies are necessary to guide us in initial treatment decisions and often to monitor treatment responses. But of course, because it's invasive procedure, it has also accumulated neurological complication risk. So with the MIN trial, we aim to develop a fast, repeatable and non-invasive metabolic MRI protocol to detect two important aspects of metabolic reprogramming, the Warburg effect and the phospholipid metabolism. Warburg effect is actually a marker for cancer cell survivor and tumor growth. And there is, you see two different types of cell, and for me, is right side is the normal cell and the left side is the tumor tissue. The most important to know about the Warburg effect is that there is a shift from oxidative phosphorylation, which happens in normal tissue, to aerobic glycolysis, even there is oxygen. And phospholipid metabolism refers to cell membrane turnover, which is a sign of malignancy or aggressiveness. We will use in this MIN trial two complementary metabolic MRI protocol at 7 Tesla, the deuterium metabolic imaging and the phosphorus spectroscopic imaging. Deuterium metabolic imaging, deuterium is actually a radioactive isotope of hydrogen with a low natural abundance of 0.01. Oral deuterium or deuterated glucose DMI is able to detect deuterated labeling of lactate, glucose, glutamate, and glutamine in the brain. And also DMI is able to generate specially resolved metabolic maps beyond the glucose uptake, which we have with PET scan. And this is actually the result of our pilot study. You see a picture of DMI, which starts from the 0 and time, and our resolution is 12 by 12 millimeter and a scan is 11 minutes. So you see above is the deuterium water and then the GLX labeled of deuterium and the labeled glucose of deuterium, which increase the labels over time. And the water is much more, the water signals much more because we have in our body also the deuterated water. And the phosphorus MR spectroscopic imaging is a novel MRI technique which helps to study brain metabolism and bioenergetics without use of radioactive isotopes. Phospholipid metabolism such as PME, PD is also able to be detected with this technique, and these metabolites are related to cell proliferation, which are present in conditions with high cell turnover, which is cancer specific. So with this metabolic MRI protocol, we aim to characterize glioblastoma metabolism and metabolic alterations during treatment. And this will help us to differentiate between treatment-induced necrosis versus tumor progressions. You heard also in previous presentations a lot about these two, but actually to not be able to differentiate between these two phenomena will cause either an overtreatment of patients or in early discontinuations of treatment. So this is actually the overview of our trial, the main trial. We will include about 20 patients diagnosed with glioblastoma, and they will receive 3 times the metabolic MRI prior to the chemo radiation, after 9 weeks, and after 21 weeks, next to the conventional MRI. And we aim to characterize the first, before chemo radiation, characterize the brain tumor metabolism, and during the treatment to assess metabolic change and response to therapy. So thank you. Is there any questions?